Hello, my name is Minseok from Seoul National University Hospital, Department of Nuclear Medicine. We are honored to share our work with the readers of Korean Journal of Radiology. This Phase 1 clinical trial of PSMA targeting agent Gallium-68 (NGUL) was conducted under supervision of Professor Chang. As a disclosure, this work was funded by Selvian Company, and this work was supported by the Technology Innovation Program funded by the Ministry of Trade, Industry, and Energy, and National Research Foundation of Korea grant funded by the Korean government. PSMA is a type II transmembrane protein overexpressed in prostate cancer cell, and it is reported that 100 to 1,000 times overexpressed compared with normal prostate epithelial cells. After a PSMA targeting ligand binds to PSMA, internalization occurs and it is retained in the cellular component. As a result, increases the deposition of a conjugated radioisotopes into the cell with subsequent improvements of both imaging and therapeutic efficacy. Comprehensively, PSMA has gained significant interest as an attractive target for the diagnostics, where this diagnostic we select a patient through a companion diagnostic image and perform targeted treatment, replacing them with a therapeutic radiopharmaceuticals, such as lutetium-177. Currently, two PSMA targeting agents have gained FDA approval. Gallium gozetotide, also known as gallium PSMA-11, is the first drug approved by the US FDA as PET imaging agent. Second FDA-approved drug is fluorine-18 labeled piflufolastat, with brand name Pylarify and more well known as DCFYL. These agents have proven that they can identify cancer that is often missed by current standard of care imaging techniques. Our group, led by Professor Jeong, has developed a novel gallium-68 labeled PSMA targeting agent based on glue-urea lysine derivatives conjugated with a NOTA chelator named gallium-68 NGUL. In a head-to-head comparison study with gallium-68 PSMA-11, this NGUL exhibited lower uptake in normal organs, rapid body clearance, and similar lesion detection ability. So to further investigate the clinical feasibility and as a required step for clinical approval, we have conducted a Phase 1 clinical trial of gallium-68 NGUL. The primary endpoint of this study was to evaluate the clinical safety and organ biodistribution-based radiation dosimetry of gallium-68 NGUL. The secondary endpoint was to show the lesion detection rate of the NGUL compared with conventional imaging modalities such as bone scintigraphy and CT scans in prostate cancer patients. We have designed an open-label, single-arm clinical trial with two cohorts comprising 6 healthy volunteers and 6 patients with prostate cancer. For the healthy volunteers, we evaluated the safety and normal organ distribution and dosimetry. For prostate cancer patients, we assessed the safety and lesion detection rate. And prostate cancer patients confirmed through tissue biopsy, and those who met one of the following conditions were enrolled, and all the patients enrolled in our clinical trial met the second condition. For healthy adults, PET-CT scans were required at 5 time points after administration of NGUL. For patient, images were acquired at 1 hour and 2 hours after injection. In healthy adults, the volume of interest for normal organs were drawn and activity was estimated at each time point. After, time activity curves were acquired and the subsequent residence time and organ doses was calculated using Olinda EXM software. Finally, the effective dose was calculated according to the ICRP 103. In prostate cancer patients, firstly, lesion was defined as any soft tissue or skeletal lesion with a focal uptake that was higher than the blood pool and not explained by physiologic uptake. All scans were reviewed separately by nuclear medicine physicians, blinded to patient's medical history. In case of inconsistency, a consensus was reached later. For the quantitative analysis, up to a maximum of 5 lesions and a maximum of 2 lesions per organ, with the most intense tracer uptake was estimated for each patient. All healthy volunteers and patients tolerate the test well, and no drug-related adverse events were observed. From the initial time point, gallium-68 NGUL is rapidly removed from the blood and showed the highest uptake in the kidney, followed by the salivary gland, lacrimal gland, liver, and spleen at 60 minutes after administration. Based on the evaluation of total body activity, up to 30% of injected activity was cleared through the urinary system within 3 hours. Accordingly, the urinary bladder wall was estimated to have the highest observed dose, followed by the kidneys and salivary glands. The calculated median effective dose was 0.025 millisievert per megabecher. The effective dose was also relatively higher than that of PSMA-11, according to the package insert. Still, the level of radiation exposure by the NGUL PET-CT examination is reasonably low, at a level of approximately 3 to 4 times the annual natural background exposure. Both adequate hydration and frequent bladder emptying can further lower radiation exposure and urinary tract uptake. Overall, 229 suspicious lesions, which is 13 hepatic and 216 bone lesions, were identified using either the NGUL PET or conventional imaging modalities. The detection rate of NGUL and conventional imaging, mostly the bone scintigraphy, were 86%, 86.9% and 49.8% respectively. However, the tumor detection rate and tumor SUV max differed according to the patient characteristics. So patients with high PSA levels, which is patient 1 to 3, showed a higher overall detection rate and tumor SUV max compared with patients with lower PSA levels. This difference is consistent with well-known results that the PSMA-avid tumor burden is significantly associated with PSA levels. And from the analysis using the Spearman rank test, we also found a significant correlation between the tumor SUV max and PSA level. This is a representative gallium-68 NGUL PET images and bone scintigraphy images of 80-year-old metastatic castration-resistant prostate cancer patient with PSA level of 61.4 nanogram per ml, showing numerous metastatic bone lesions in the NGUL PET. However, limited visualization of lesions in the bone scintigraphy. The quantified uptake in the metastatic lesions between 60 minutes and 2 hours after NGUL administration demonstrated a significant increase in SUV max and SUV mean by approximately 15%. However, the tumor-to-background ratio defined by the blood pool uptake as a background showed no significant change between 60 minutes and 2 hours. So considering the practical issues, we recommend scanning the patients at 60 minutes after the NGUL administration. So in summary, gallium-68 NGUL was well tolerated without demonstrable drug-related adverse events. And this NGUL novel PSMA targeting agent distributed along the kidneys, salivary glands, lacrimal glands, and rapidly excreted through the urinary system. So accordingly, the highest observed dose was found in urinary bladder wall with an effective dose of 0.025 millisievert per megabecquerel, which is reasonably lower compared to the natural radiation. And the lesion detection rate was higher compared with conventional imaging modalities, especially the bone scintigraphy. And according to the quantitative uptake and tumor to background ratio, we recommend scanning the patients at 60 minutes after the administration. Thank you very much for listening. And for more information, please refer to our article.